This is Module 2, Section 3. This lesson reviews the visual system. So we've already gone through the auditory system, and next we will be doing the tactile system. So we know that the auditory system works according to a tonotopic map, which means that the various receptive areas are arranged by frequency, and we'll see a similar concept at play in the visual system. So our key questions for today: what is the visual cortex, how is vision processed, and how can vision be restored? So the visual cortex is located in the occipital lobe. It takes inputs from the optic nerve, which run from the eyes to the visual cortex. And it outputs to other cortical areas. There are two pathways. There's the ventral pathway, which takes information from the visual cortex to the temporal lobe, and it's used for object recognition. And then there's the dorsal pathway, which takes information to the parietal lobe, where it is integrated for visual motor processing, because obviously movement and vision have a lot of feedback processes associated with one another. Every step in these pathways is organized according to a retinotopic map, meaning that the pathways of cells are organized according to locations on the retina. This means there's a corresponding area on the primary visual cortex for areas on the retina. And just like we talked about in the lesson on biological neural networks, the visual cortex is thought to be organized in layers. So we have V1, which takes in unspecific information such as loose shapes, maybe lines or curves, and then V2 takes in a bit more specific information, color, texture, and then as you go higher up in the visual cortex, the features that are identified become more and more specific. And that's just something that we talked about in biological neural networks, and you can see here in this image. So, of course, the signal that we're processing is light. It starts from light. It starts from photons. So we need some way to deal with that, and that is the eye. So the cornea and the lens focus light onto the retina, which is the back of the eye. The pupil acts as an aperture by constricting and relaxing to control light levels. And the retina is the tissue, and the retinal pigment epithelium contains highly pigmented cells. The retina contains a pit called the fovea, which is specialized to contain the highest acuity vision due to less cell layers. The special cells that process photons are rods and cones, and they're intermixed in the retina. At each position, there are these photoreceptors that are detecting dim light, which is rods, or bright light, which is cones. They're not uniformly distributed, which kind of helps explain the retinotopic map and why there needs to be specific areas for, or specific pathways for specific areas on the retina, is because the constitution of each area is mixed with rods and cones, for the most part. But there are a few parts that are specific. So the fovea only has cones, and the periphery of the retina is very full with rods, because peripheral vision is more light sensitive, which enables you to see dimmer objects in your peripheral vision. So now we get to see these rods and cones. Rods are long, cylinder-shaped cells that appear to have free-floating stacks of disks within the membrane. They're sensitive to low levels of light and are therefore responsible for vision under dim lighting. Cones are responsible for vision in regular light, and the disks are continuous with the outer membrane. Cones can be further categorized by the wavelength of light that they respond to. There are long-wave cones, medium-wave cones, and short-wave cones. It's the integration of these wavelength-specific signals that allow for color vision, as different colors correspond with different wavelengths. Upon reception of a photon, the membrane of the photoreceptors is actually hyperpolarized, which is different than other cells, and through a series of biochemical events, that signal is transformed into vision. And these biochemical events in total are referred to as Phototransduction. So this is the conversion of light to hyperpolarization. In the dark, rods are depolarized steadily to release glutamate. And so, of course, the release is continuous and it's proportional to the membrane potential. Light hyperpolarizes the membrane and decreases glutamate release. And of course, the brighter the light, the stronger the hyperpolarization. And yes, just another note about where these photons are absorbed. They're absorbed in the outer segment, right? So that's kind of this area. Okay, look at visual processing as a whole. From the photoreceptors through the retinal ganglion cells, from the photoreceptors, the signal moves through the retinal ganglion cells whose axons make up the optic nerve. There are two types of ganglion cells, on cells and off cells. On cells decrease firing when an object is dark, and off cells increase firing when an object is light. This specificity allows for integration of information before the signal reaches its next destination, the lateral geniculate of the thalamus. Remember that the thalamus is the hub of information in the brain and has projections to all major areas of the cortex. From the thalamus, the signal is sent to one of three pathways in the primary visual cortex. M cells take information about movement, P cells take information about spatial resolution, and K cells take information about color. Once the signal has been processed in the visual cortex, it is sent in either the ventral or dorsal stream. The ventral stream projects to the temporal lobe, as I said before, and the dorsal stream projects to the parietal lobe. So in recent years, scientists have crafted an artificial retina. There was actually one that was approved by the FDA in 2013. And these are to replace destroyed photoreceptors. So in a similar way to how a cochlear implant replaces a damaged basilar membrane, an artificial retina would replace destroyed photoreceptors. It's a small chip with many electrodes that are connected to a camera that rests in front of the eye. Information from the camera is encoded and sent to the chip, which in turn activates the retinal ganglion cells or the optic nerve and allows for some form of vision restoration. One of the most difficult issues with this piece of technology is figuring out how to get it to not overheat, because obviously the tissues around your eyes are very sensitive. And artificial retinas are still very new, and they're highly uncommon, but this is just an area of research in restoring vision. So, quick summary. Vision processing starts with the photoreceptors in the retina and follows retinotopic pathways through the optic nerve to the thalamus and then to the visual cortex. From the visual cortex, information is sent to the temporal and parietal lobes, and damaged photoreceptors can be replaced by an artificial retina. In the last lesson of this section, we will be talking about tactile sensation.